Alright, so we're picking up where we left off, which was phagocytosis. So we're going to watch two videos that are here. One of them is the neutrophil chase. This happens to be one of my favorite ones. Hopefully, oh, let me see. Let me do it big.
Neutrophils are white blood cells that hunt and kill bacteria. In this spread, a neutrophil is seen in the midst of red blood cells. Staphylococcus aureus bacteria at the end. The small clump of bacteria release a chemoattractant that is sensed by the neutrophil. The neutrophil becomes polarized and starts chasing the bacteria. The bacteria, bounced around by thermal energy, move in a random path, seeming to avoid their predator. Eventually, the neutrophil catches up with the bacteria and engulfs them by phagocytosis.
So it literally just took somebody's blood. Yeah, sure. Thank you. Pause button. Okay. I hate that. Okay. That is scary looking. Okay, moving on.
These human neutrophils, taken from the blood of a graduate student, are mobile cells that will quickly migrate to sites of injury to help fight infection. They are attracted there by chemical signals that are released by other cells of the immune system or by invading microbes. In this experiment, tiny amounts of chemoattractant are released from a micropipette. When neutrophils sense these compounds, they polarize and move towards the source. When the source of the chemoattractant is moved, the neutrophil immediately sends out a new protrusion, and its cell body reorients towards the new location.
So they remind me of a protozoa that kind of moves like that. Let me hit the right pause button. Anybody seen in your biology class maybe, or in my microbiology class, a eukaryote, a protozoa that moves like that? Amoeba. Doesn't it look like an amoeba? Right? It's pushing its cytoplasm, right? And it literally is engulfing, just like an amoeba does, right? An amoeba sends out its cytoplasmic extensions and engulfs its food. Our cells do that same thing, right? Our phagocytic cells, that process of basically how an amoeba eats. And in fact, let me shrink this, right? So, amoeba eating paramecium. And of course we're going to get an ad. Yeah, I know. There we go. Oh yeah, I forgot. This one has horrible background noise. It's like some big class. Right? So you see how it's just taking its cytoplasm and it's engulfing it? And it actually takes it into a vesicle. Right? So these two paramecium, and then they're dumping digestive enzymes in. So these poor paramecium are trapped, right? They're trying to get out, they can't get out. And eventually they're going to be digested by the enzymes. So then they just become a blob over time. Poor paramecium, right? So we didn't see it in this great of a detail because it's much smaller, right? But I don't want it to play the next one. That's the same thing that happened with the bacteria, right? They got engulfed into a vesicle by that neutrophil, and they got digested and killed. Okay, and that neutrophil, that particular type of white blood cell, how did they say that they knew that that bacteria was there? Yeah. There were substances released by the bacteria, right? What they call chemoattractants. So they're chemical attractants. That bacteria was a non-motile bacteria, but your blood has liquid, right? So it was being bounced around by thermal energy. But there are some that are motile, right, that could kind of swim away, and that's an advantage that some bacteria have, is they could swim away from the neutrophil. But if it's not in an aqueous type environment, they're not going to get very far, right? You can't swim without water, right? And the same thing holds true for those flagella. But so we have cells that can do this process that we know as phagocytosis. One of the ones that really specializes and does a lot of that is the neutrophil, right? And these white blood cells are found in your blood. So they literally just took a sample of blood, put it on a microscope slide, stuck some bacteria on there, and filmed it, right? Because that was what will naturally happen. These cells can detect pathogens, bacteria, and they will go after them, gobble them up, and destroy them by phagocytosis. And they're very much, as you saw in the second video, they're going towards those attractants, right? Those chemical attractants that are given off by the bacteria, they can sense it and they're going to head towards it. Plus, our body is going to release stuff. Remember when we had inflammation and our cells are damaged? We send out chemoattractants, right, that are going to attract those neutrophils to that area, right, and also macrophages, these other specialized white blood cells. But both of these guys can do phagocytosis. So for phagocytosis, the very first step is that chemotaxis, that chemical sensing movement, right? Chemo meaning chemical, taxis movement, that chemical movement to that area. Then once they get there, they have to recognize the invader and attach to it. So think about these steps, right, as we watch the animation in a moment. What is causing the chemotaxis? They're going to name a particular molecule. What is allowing them to recognize? They're going to name a particular molecule. How are they able to attach, right?
The antibodies?
Nope. Wait, not there yet. Okay? And then they're going to engulf it into a vesicle, right? What's the name of that vesicle? That vesicle is going to mature into a phagolysosome, right? How does it go? What is that maturation process? Then what's inside there gets digested, right? It's destroyed, it's digested. And then it's going to leave the cell by exocytosis. How does that happen? Right? So think about each one of these steps as I play the animation, and then we're going to go over it. Oops. I had muted it.
In phagocytosis, phagocytes are attracted to the area of invasion by chemical products of the microorganism, phospholipids released by injured mammalian cells, or by components of the complement system. The phagocyte moves into the area of invasion and then attaches to the microorganism. The attachment is mediated by a variety of surface receptors, including antibody, lipopolysaccharide, and complement receptors. For example, the C3b component of complement coats bacteria or other particles and then binds to C3b receptors on the phagocyte. This process of coating to enhance phagocytosis is called opsonization. The microorganism is then engulfed by the phagocyte into a vacuole known as a phagosome. Vesicles in the cytoplasm called lysosomes fuse with the phagosome, releasing digestive enzymes such as lysozyme and proteases into the phagosome. The structure resulting from this fusion is called a phagolysosome. Inside the phagolysosome, microorganisms are killed and digested. Finally, the digested contents of the phagolysosome are eliminated from the phagocyte by exocytosis.
Alright, so now we're going to go through each step. Alright, so this is a diagram that has all those steps, right? 1 through 6. We're going to go stepwise through it. So for this diagram, right, chemotaxis is phagocytic cells recruited by chemoattractants. So chemoattractants are products of the microbe. So they can be phospholipids, or they can be things from injured host cells, right, when we have injury. The chemical substances, the group name we give to ones that are involved in this movement to the area are called chemokines. In this diagram, C5A is listed as a chemotactic agent, right? It's a chemoattractant. It's causing the phagocytic cell to head towards this microbe. This C5A is a complement protein, something we're going to learn about later. And that's what this C here stands for, is complement. Now, for recognizing it in attachment, right, how were they able to recognize this bacteria? Yeah, with their receptors. Their receptors are for what molecule? C3b. You see C3b? And then the receptor binds to the C3b. The C3b is produced by our bodies and basically tags the bacteria for destruction. What name did they give to this process where our proteins bind to it and therefore we're going to destroy it? It started with an O. Opsonization. Good listening skills. Opsonization. Basically means tag for destruction. And so since this is another C and then a number and a letter, what do you think that first C stands for? Complement. Right? This is a series of proteins found in your blood and in your tissues during inflammation. So as you can see, they're really helpful, right? One of them is going to help get the bacteria to that area. In addition, that bacteria itself may cause these guys to sense and move into the area, or our own cells that are damaged, or our own cells that are releasing chemokines, chemical attractants to our phagocytic cells. So then they mentioned other things, right? What else can a phagocytic cell recognize that's maybe bound to a bacteria? You said it, Tyler.
Antibodies?
Antibodies, right? If we have antibodies that are binding, again, our phagocytic cells have receptors for our own antibodies. And then we have a whole group of receptors referred to as toll-like receptors. So these are receptors on these phagocytic cells. So this isn't the only type of receptor they have. C3b is only one of a bunch of different types of receptors that we have. But the important thing is that we have to be able to recognize this as a bad guy and literally attach to it in order to then engulf it, right? And form that vesicle. And what's the name of that vesicle? When we're doing phagocytosis, it's a phagosome, right? And again, we want to keep the bacteria or the virus or whatever it is that we engulf, we want to keep it in a membrane-encased vesicle called the phagosome. So just like we saw with the amoeba, right, the pseudopods, we're going to surround it and we're going to form that vesicle that we call a phagosome. And then lysosomes, which are endosomes, right? So they're vesicles inside. They're going to lower the pH. They contain digestive enzymes. When a phagosome and a lysosome fuse, what becomes the name of that vesicle? Phagolysosome. Bridget, I already posted this, or you can't see. Okay. Okay, so it's a phagolysosome. So what did they basically do to the name? They just fused the two names, right? Phago for phagosome, right? And lysosome. They just fused the two names together. right? Because that's really what's happening there too, right? A phagosome is fusing with a lysosome. So that's kind of cool, right? They just fuse the two names together. But what is happening here is those lysosomes contain digestive enzymes, right? They're going to lower the pH. And why? Why do we want to do that? We want to destroy, right? Our next thing is what's going to happen in there. We want destruction and digestion of whatever it is we engulfed. And then the last step is that membrane itself is going to fuse with the actual membrane of the cell and discharge any extra stuff. So in that digestion, right, there are toxic reactive oxygen that's produced, uric oxide, the pH decreases, right, so it becomes acidic. Enzymes degrade. Defensins, which are molecules that we produce that literally poke holes in the membranes of invaders. Lactoferrin is a protein that we produce that ties up iron, because especially for bacteria, they need it for their metabolism as well. And we want to secure iron, keep it for ourselves.
Does this go back to like with the holes, the fenestrations, and the different type of holes in biology? Like in having.
Oh, you're talking about in your capillaries?
Yeah.
No. Completely different. So, okay. So cells that do phagocytosis, your two main phagocytic cells, right, they're gonna gobble stuff up and digest it like this, are your macrophages and your neutrophils. So these guys have in that's similar, that's the same as they both do phagocytosis. Your macrophages are always found in your tissues. Your neutrophils, for the most part, are only going to go there during inflammation. They're part of the cells that get called to that area when you have the inflammatory response. Your macrophages are known as monocytes in your blood, right? So if you take somebody's blood, right, and you look at their white blood cells under the microscope, those of you guys in anatomy and physiology 2, anyone in 2? AMP2 or have taken AMP2, right? You remember monocytes, right? The really big cells, the really big horseshoe-shaped nucleus. These guys are what become your macrophages in your tissues, right? So they go through a little bit of a differentiation, a little bit of a change to become these basically super eaters. Macro meaning big, right? They're big eaters. Neutrophils are in the group that we commonly refer to as granulocytes because their lysosomes, right, inside of them, kind of stain. They actually neutral stain. That's why they're called neutro, right? Neutrophils.
That's what granulocytes again?
We'll get to it. You don't have to write it now. I don't want to goof it up. So all your granulocytes, you'll notice all their names end in "phil," right? And they all have, before that, some indicator that actually has to do with those granulocytes, those vesicles, how they stain under the microscope. So for neutrophils, there's kind of neutrally stained. Like you can't really, unless it's a really good stain, you usually can't differentiate their granules from their cytoplasm. Unlike basophils or eosinophils, their granules stain quite distinctly. Eosinophils, their granules stain kind of a reddish to orange color. This is because they're stained by the dye called eosin. This is where they get their name, eosinophils. Basophils, their granules stain with the basic dye. That's where they get their name, basophils, right, for basic staining. Okay, so that's where they get their names, right? So these are two relatively different from each other cells, but they're going to do similar jobs, but a little bit differently from each other. So they both do phagocytosis, but your macrophages are always in your tissues, right? So they're the guys on guard, right? But when you have inflammation, we're going to call even more big eaters in. And who aren't normally there, which is your neutrophils, they're going to come too, right? And they're eaters too, but they do this a little bit differently. So macrophages can survive after phagocytosis. They can basically keep making those lysosomes. They can keep making phagosomes and lysosomes and keep digesting and eating. So by contrast, do you think neutrophils do that? No. Once they use up their granules, right, those vesicles, those lysosomes, they die. So my nickname for these guys is your suicide bombers, right? Because they are very powerful killers, but pretty much once they drop their payload, that's it. They're done, right? So they will actually kill themselves in an effort to contain the infection. So hence the name suicide bombers. They will kill themselves in the bombing effort to try and keep the bad guys in that area. So hence why we really don't want them always in our tissues, right? They're a little crazy, right? They kind of sometimes get out of hand with the bombing and make a mess, right? We don't want them uncontrollably going crazy all the time, right? So we really kind of control these guys. We really kind of only call them in when they're necessary. Does that make sense to you guys? All right. Macrophages, although neutrophils start out like the serious killers, macrophages can increase their killing power if they become what's called activated by T cells. So these are specialized lymphocytes that can communicate directly with the macrophage. And basically what happens here is the T cell recognizes the specific invader and can help the macrophage determine which toxic chemicals it should try next, right? Because it has this whole arsenal of stuff, right? Uric acid and reactive oxygen and different enzymes. And so it may change up basically the different enzymes or different toxic substances based on this information it gets from the helper T cell, from this communication, and therefore increase its killing power. Neutrophils, right, have, as I said, the greatest killing power right out of the box. Macrophages can fuse with the help of the T cells into giant cells, and then with involvement of T cells into big masses of cells that we refer to as granulomas. Neutrophils don't do this. No. This would happen in animals as well. Any animal that has an immune system, which is all animals have an immune system. When you get to invertebrates, they have certain components of the immune system we have. They don't have the adaptive components that we do. That's true. I guess invertebrates are considered animals though, huh? Right. So for vertebrates, yes. Invertebrates, some of this. Definitely this component though. They're not going to do the high-tech stuff we'll get to in adaptive, like making antibodies. Good question. Okay, so for neutrophils, they're not going to fuse together like macrophages to form giant cells or anything like that. But as I said, these are your suicide bombers. They will literally kill themselves to try and trap the bad guys in the area. They will shoot their DNA out of their cells, kill themselves, to create a net out of their DNA, right? DNA is a huge molecule. It's bound up with a lot of protein. If you release those proteins, right, you have these huge molecules that can basically be used as nets to trap in the pathogens in that area. Pretty cool ability that they have. So when we talk about the white blood cells, we really want to home in on functionality. What is it can they do, right? So we've already talked about neutrophils, right? They're going to phagocytize and digest material. We also mentioned the macrophages, which remember are just differentiated monocytes that are in our blood. All of these guys phagocytize. So do the dendritic cells, although notice for their function, they don't really mention that. But this whole collect antigen, that's how they collect it, is they engulf it and they break it down into pieces. But they don't just stop there. They use those pieces to present this information to your lymphocytes. And this is that bridge to your adaptive immune response. And as it says here, right, the very short explanation of their function, it just says adaptive immune response. And they're really only showing 2 lymphocytes here, but truly you have 3 types of lymphocytes. But probably the reason why they're only showing 2 is because only 2 of them are involved in your adaptive immune response. The other one is not. Does anybody know the name of your lymphocytes? They're commonly abbreviated with letters. B lymphocytes, T lymphocytes, and then the other one is,
See?
you got it, NK, natural killer cells, right? And it's the natural killer cells that aren't part of the adaptive immune response. It's the T cells and the B cells. And it'll make sense why they have those names when we go into their functionality a little bit more specifically in their lineages. But those of you guys that have taken anatomy and physiology, right, under the microscope, you just called them all what? Lymphocytes, right? Because you cannot tell with the stain that we do for just a normal what's called the differential, differentiating the major groups of white blood cells under the microscope. We don't go to the level of what type of lymphocyte is it. Is it a B cell, is it a T cell, or a natural killer? We just go to the level, is it a lymphocyte? I also caution students not to use the term leukocyte. What does leukocyte mean? White blood cell. What does lymphocyte mean? It's either a B cell, T cell, or a natural killer. A lymphocyte is a very specific group of white blood cells. And beginning students a lot of times confuse leukocyte with lymphocyte. This is a big no-no because leukocyte is a huge term. It means all of these guys, right? All of the white blood cells. So I always say if your A&P 1-2 instructor will let you get away with it, just call them white blood cells, right? So you don't get confused. And it's easier to spell in the practical exam, right? Than leukocyte. And I will typically always refer to them as white blood cells. It's pretty straightforward. We usually don't screw it up, right? But you'll hear sometimes people say leukocyte, and they mean white blood cell. And they're talking about all your white blood cells. They're not being specific. And as you can see, and here's your granulocytes, Tyler. Granulocytes. There are 2 main groupings that we typically put cells in, what we call granulocytes and agranulocytes, meaning that they don't have granules that stain. So your granulocytes, what they have in common is that they have granules that stain. Now, granted, neutrophils, as I said, their name comes to the fact that their granules don't really stain all that different from their cytoplasm. So they're really kind of hard to see. Eosinophils are named after the fact that their granules stain with the dye eosin, so they're going to be like a reddish to orange color. Basophils and mast cells are found in your tissues. This wouldn't be found in your blood. Basophils can be found in your blood. These guys stain similarly, they have granules that stain with the basic dyes. And that's why they're called basophils. So again, the other thing that helps you remember too is all their names are end in "phil," right? Because they're all filled with granules that stain using the staining procedure we use. Depending on what color and whether you can see those granules or not determines which one it is. So why do you think their granules stain differently? Why do these vesicles stain differently? Because they're what? Are the vesicles the same? No, because then they'd all stain the same, right? So clearly they're different, right? There are different substances in the granules or these vesicles inside neutrophils. There's different stuff inside eosinophils. There's different stuff inside those vesicles for basophils. So is their functionality different? Absolutely. These granules in eosinophils participate in inflammation reaction and immunity to some parasites. Right? So parasitic worms, a lot of times when individuals have that type of infection, you'll see an increase in eosinophils, right? Because the substances that are in those granules are effective against that type of pathogen. Basophils and mast cells, not my favorite cells, because they release things like histamine and other inflammatory inducing chemicals from their granules. And so these guys are a lot of times involved in the allergic response, right? Hence why I don't like them so much. These unfortunately get triggered when people are allergic to things.
What is their function just for allergies? What about the inflammatory process? Is that still considered the same?
Actually, eosinophils are they're kind of the polar opposite of basophils. The things that they release kind of shut down inflammation. So it says participate in inflammatory reaction, but truly they stop the inflammatory reaction. So again, when they order what's called the differential, what they'll do is either a scientist like myself or a machine will count 100 white blood cells and categorize them in these categories. Is it a neutrophil? Is it an eosinophil? Is it a basophil? Is it a monocyte? Because remember, this is the one you'll find in your blood. Or is it a lymphocyte? These 5 types. And there's a 6th. Neutrophils, when they're really young, can be identified from neutrophils that are mature. The difference between young and mature ones is their nucleus. So that's another common element that usually runs in the phils, especially with your neutrophils. What do you notice about its nucleus? It's kind of morphed, right? See how, like most nucleuses, we're used to seeing this nice round thing, right? Or relatively round. This one has an indentation. See how this one's stretched out? It's what we call lobed or multi-lobed. So that's why sometimes these cells are referred to as polymorphonuclear. They're referring to that there's many morphologies to the nucleus. Right? And so sometimes it's even abbreviated PMN, which stands for polymorphonuclear. Polymorphoneutrophils.
And how can you tell the difference between the mature and
Yeah. So the mature ones are going to have that polymorphonuclear shape, right? Where that you have that multi-lobe, stretched out, funky looking nucleus. Where the immature ones, the nucleus tends to be stretched out, right? So something like you see here for the eosinophil, or maybe even more compact like you see here for the monocyte. So it hasn't matured to the level where the nucleus has morphed. This is not a good sign. You don't want to see these guys usually. You don't want to see immature neutrophils in the bloodstream. So that's one of the first things that they look for, right, is look at its nucleus. If it's a multi-lobe nucleus, then the next thing they look at is what do the granules look like? Can I see them? If you don't really see the granules, you know you're looking at a neutrophil. If you can see the granules and they're reddish, it's an eosinophil. And then if you're lucky enough, and the reason why I say that is that these guys are very rare. Basophils are mostly pretty rare. And their granules are going to be like a purplish blue, which is also the same color as the nucleus. So sometimes it's really hard to see. It all seems like a jumbled mess. Now these are similar colors to what a lymphocyte would be. So this is where magnification really matters too, in really detailed looking at your cells, because lymphocytes are going to be a nice big purple nucleus. And if you look at that nucleus under high power, it'll almost look granular. The trick to know that you're looking at a lymphocyte is you're going to look for a very thin layer, hopefully, of light blue cytoplasm. Then you know it's a lymphocyte. And chances are, if you're looking at it under the microscope, y'all, you're looking at a lymphocyte. Because, again, basophils are very rare. Of all the few years that I taught A&P2, I think I've maybe truly saw 1 or 2 basophils. They're that rare. You see Miss Erica do her happy dance. You get all excited, right? So I always caution my students that have to take A&P2, draw out what you see and argue with your instructor. Because if they say it's a basophil, I seriously doubt it is. I seriously doubt it. Right? So what's the saying you guys learned? Never let monkeys eat bananas. Never stands for neutrophils, because those are the most abundant, right? That's what you're going to find in normal blood very easily. Right? With infection, you're going to find even more of them, right? Because we're going to call them up out of the bone marrow. So never let, L stands for what? Lymphocytes. So again, those are going to be quite numerous in circulation. You're continually producing them all the time. Monkeys stands for monocytes, which is going to be the next abundant in your blood. Eat eosinophils, right? And they're going to be very low, right? But you should be able to find them. And then very last is bananas, which stands for basophils, which are extremely rare. Right? Out of 100, you might find one. Might. Chances are you won't. 0. Out of 100, you should find 2 to 4 eosinophils. Out of 100, you should find 55 to 65 neutrophils. Right? Those are really easy to find. Right? Out of 100, you should find 25 to 35 lymphocytes. Again, should be pretty easy to find. 3 to 8% are going to be your macrophages. And these guys are really easy to find too, because they tend to be really huge. They're double the size of a red blood cell. Where your lymphocytes are usually going to be around the size of a red blood cell. So under the microscope, they're my favorite thing to look at. I just think they're absolutely beautiful. And when you learn about what they do, right? They have such awesome abilities. It's impressive.
Do they save the slides?
In the A&P lab, they're prepared slides. So yes.
Like in hospitals, because I actually had some basophils, but I wanted to know if I could ask them. Do they save the slides?
Well, it depends on whether they did a slide or used a machine. There are machines now that can do this. Yeah. Yeah. A lot of times a machine is doing it. I used to do them by hand. But a lot of times they're done by a machine. So that's it for phagocytosis. So let's